Morning, Catherine.
Good morning. Thanks, Nicola. And yes, good morning, good afternoon, good evening, depending on where you are. But I just wanted to wish you all a very warm welcome to this workstream showcase from Drug Substance. This is the first time that we've run workstream showcases, and this is we did have a go yesterday, so this is the second one. So we're learning a lot as we go, but very excited about the workstream showcases, whether you're new to drug substance or whether you are already in a workstream and want to, or involved with BioPhorum and want to know more. I think this will be a great opportunity to learn a bit more about the drug substance programme. And as you can see, we've got 4 of these planned for this year, and by the time that we get to December, we will have covered everything in the drug substance programme, ready to go again in 2025. Your feedback is incredibly valuable to us, particularly as we are mastering the technology. So if you do have any suggestions or comments, please either drop them into the chat or feel free to email any of us afterwards to let us know, as we, in the spirit of continuous improvement, we want to make these as good as we possibly can. That's all that I want to say. I think it's time to get on with the presentation. So I'm going to hand you back to Nicola and the team, and hope you enjoy and find this session informative.
Thank you very much, Catherine. So just giving you a little bit of an overview what we have planned for this session. So the idea for each of these quarterly work streams is that we will workstream showcases, is that we will feature 3 of our drug substance work streams. So for today, we have a little bit for you on the making and moving operational cell banks work stream, multi-product facility, and this includes the protein A resin reuse sub-team, and also LIVCA. But I will start us off just with a little bit of an overview about BioPhorum and drug substance. So firstly, I'm Nicola Powell. It's a pleasure to see you all on the call this morning. I'm an account manager within drug substance. We know that we've got sort of mixture of attendees along to these sessions, and it's possible that some of you have not been involved with BioPhorum before. So just to give you a little bit of background, BioPhorum is really a unique collaboration for the biopharmaceutical industry. We're able to bring together leading SMEs from across the globe, across many different member companies, to tackle some of the most pressing and challenging issues we have in the industry. And the idea being we have greater power to advance the industry and influence the landscape as this collaboration than any one member company can alone. Within BioPhorum, we are arranged in different forums depending on different areas of interest within the biopharma industry. We are representing the Drug Substance Forum, the team here today, and there's a little bit of detail on this slide to give you a bit of background on drug substance and our mission, and how we're looking to introduce work streams and bring together SMEs to accelerate technology adoption, implement post-approval changes more rapidly, and increase confidence in maintaining compliance, all really to advance excellence in drug substance manufacturing. So going back to this session and what it's about, they really are high-level introductions basically for our work streams. So equally suitable for SMEs that are perhaps looking to understand a bit of background, considering getting involved. For our drug substance leaders, as a bit of an overview on what the different workstream are, what they are, and where they're up to. And also, these sessions are completely open to anyone from our member organisations. You don't have to have been onboarded and know about BioPhorum. So even if there's just a topic that you hear we're working on and you'd like to know a bit more, these sessions are hopefully useful for you. Our intent is for them to be relatively informal and interactive. We're quite pleased that we have now ungagged the audience and you have the ability to unmute yourselves and join in. Please do. We'll probably run each of the workstream presentations and then take a pause for any questions. So by all means, feel free to pop questions in the chat or raise a hand and we'll take it from there. We've allowed 90 minutes. We were a little under that yesterday, so it's likely that we will be able to give you a bit of time back at the end. But the important thing is that the space is there for the questions and the discussion. So don't feel like you need to hold back if there is something that you would like to ask. And we are recording today and we will make the slides available after the call. So I think there's probably quite a lot of links and things in the slide deck. This will all be sent on to you afterwards. If I just one more slide from me and introducing drug substance. So this one gives you just a bit of a high level overview of our different work streams. So the top section, top row here lists out the work streams that are currently active in the Drug Substance Forum. You will see below that we organise ourselves in the main into different sub-teams. So you can see, I think this shows quite nicely, not all work streams are equal. Some of them are trying to tackle a huge area and you can see have therefore split themselves down into different sub-teams of SMEs to focus on specific topics and specific deliverables. We also have the communities of practice, which is worth noting. So these are in fact teams that were work streams. They have delivered and achieved their objectives. So they may have delivered a publication, for example, but for several reasons, we have chosen to keep that community, that team of experts together because they are able to track implementation of what it is that they have developed through the industry. They are able to respond rapidly to, say, new changing guidelines or other members' asks. So we have Ask BioPhorum, which is a facility for any of our participating members to put questions to the industry, to their peers, to find out, benchmark how others are doing things. And equally, the communities of practice meet at not as frequently as work streams, but they do meet on occasions through the year to sort of scan the landscape and track what could be new hot topics coming up in their particular area. And it's possible that a community of practice could in fact come back to an active work stream and work on delivery of something new. You can see on here highlighted in orange, these are the work streams that we're going to be talking about on today's call. And I will go ahead and kick us off by introducing my colleague Sean Collins, who is going to start with making and moving operational cell banks. Thank you, Sean.
Thank you, Nicola. Good morning, good afternoon, good evening, all of you. Thank you very much for taking the time to join us today. As I said, I'm Sean Collins here. I'm one of the facilitators here at BioPhorum, and I run this workstream. Now, one of the reasons we wanted to particularly bring up this one is kind of multiple reasons. One, it's a workstream it's at the end of its life, so it's just about it's transitioning at the moment into a community of practice. So it kind of shows you that process and what it's achieved and what it's done. But this particular workstream also we like to chat about it because it has a number of really good examples of what collaboration and the real socialization of the work that we create, the impact that can have, and the work that you can all what you can all achieve by working together and producing something valuable in even in quite a focused area such as this. So this particular work stream was started in 2020 with a particular focus on the replenishment of working cell banks, because what it was identified is that the guidance that existed was very minimal, and there wasn't an awful lot there for exactly what needed to be done, and it was all down to a lot of interpretation to each individual company. And so what our members thought was we could create essentially a guide on how to approach the correct qualification or the replenishment of working cell banks. So that was the goal they came out to get to there. And then, so next slide then, please. And so what they ended up doing was working on creating a white paper. And so it was really, I'd say, to focus and explain and kind of decide this is how we should go about doing it. So they created a very comprehensive paper on using the title there. The link and the title is at the bottom of the page there. You can see it's the science-based approach to qualifying replenishment of working cell banks and industry review. And what this very much is a risk-based approach to how to, depending on the change you're making, depending on what's being done, how you can qualify it and what should be achieved and done. Now, this paper was worked on and published in November 2021 by the team. It was very well received by our members and by the community. And what the team ended up doing after that was then they spent a considerable amount of time socializing it, because it's all well and proper creating this fantastic guide, but unless people know about it and unless people understand it and start using it, it's not going to go anywhere. So the team very much after that focused on getting the message out there, so sharing it internally within their companies across different parts. They then took it to conferences. So the group worked together, created a collective conference presentation, so different members could go to different conferences around the world and share what this paper is about, what we're trying to achieve, what we're trying to focus and really get some actual structure into how these should be assessed and completed. And that was, again, but everywhere they presented, it was very well received by people who were there. So it seemed like a great idea. It seemed like really a sensible approach. No real challenges to it. There was expectation that we'd try and get some regulatory feedback as well, but and just really understand that that's how whether they're going to accept it or not. And then it came down to basically the companies who are doing it actually implementing it and using it. And one of the great examples we have of this is we actually have a podcast, which again the link's in the bottom of this sheet here, with one of our member companies who was Takeda, who were influential in creating this paper. And one of the reasons that we have that there is because they successfully cited the paper as a rationale for refusing to perform additional characterization as requested by the FDA when they were going as part of their replenishment process. Now, this was quite a big deal because they'd replenished it, and the challenge back saying you need full characterization. They said, well, we don't believe we do, because we've assessed the risk based on this paper that's been published out there, and we believe that we don't need to do it. And it was accepted. So the FDA backed down, said, okay, fine, we accept that based on this risk assessment, it is an equivalent of a like-for-like swap, and therefore you don't need to do full characterization. And that's huge as far as, in particular, cost savings are concerned. I think in the podcast they talk about full characterization could cost anywhere from $500,000 up to $1 million, and so that's a massive saving, and that's just in one instance that this paper has managed to save one person. So that's a tangible number that you can put to what the value of this paper can achieve, and that's just one paper in one group being achieved by the vast amount of pieces of work being done by BioPhorum. And so it's a real example of what the collaboration and bring the whole industry's views together to create a best practice approach that can have significant impact. And it goes beyond that, because now not only is it something that you potentially use to defend and minimize the costs and work being done, it's been identified by the ICH, and the ICH now reference this particular paper as part of the training material for their risk-based decision making as part of ICH Q9R1, which is brilliant. That's an absolutely fantastic achievement, because it shows that it's been well thought out and executed. It's specifically, yeah, there you go. Thank you, Nicola. This is the slides directly from the ICH presentations. So this is all about their training on rule-based decision making. So they decided that this particular paper was a prime example of how to go about using risk-based decision making and applying it correctly. So you can see here that what they say, and then that is another slide from the ICH slide deck, and that is pulled. That's there is pulled directly from our paper. So whilst the text is quite small, you might be able to read it, but that is in the paper itself. So if you want to go back and follow the link to view it, it's freely available on our website for all to see. But that little table there is a perfect illustration of what the paper stands for. It boils down essentially the scenarios and what kind of classification, what risk you need to give it, and what kind of qualification that needs to be done. So it's exceptionally, but I say been very well received, and as I say, even referenced by the ICH. One thing that people like to often ask us is, how did the ICH find out about it? And in all honesty, we don't actually know. We don't know exactly who picked it up and who put it in there. But this is another reason why we say that it's a prime example of what socialization can do. We pushed everything, we pushed all we could with this particular paper. As I say, we had multiple members taking it to various conferences, presenting on it, sharing the message, getting it out there, what can be achieved, what can be done. We utilized it through our marketing, through our LinkedIn. We had the podcast that was created. So it was essentially a multi-pronged approach of just getting the message out there. This has been created. This is what it stands for. Everyone, please pay attention. And it's clearly worked. It's been picked up. The ICH saw it and thought, that's a great example. Let's include that. So it's something we like to shout about, because it's a real achievement for our members in particular, that they've created such a fantastic paper. And that's, yeah, thank you. I mean, no thanks to me. I say I can't take any credit. I was not involved in this work stream by the time when this was all been done, so I can take no credit for that. But I look after them now. It's all down to the members. So after that was done, it was then decided what could be next. They've achieved their goal. And the next process then is either, do we shut it down? What else do they do? The team were very keen to stay together, for two reasons. One was that the implementation of the actual paper at various sites is still ongoing. So a lot of companies are still in the process of trying to adopt the new process that's in the white paper and aren't quite there yet. So the benefit of keeping the team together means that they have that continued avenue for discussion and interaction with the rest of the members, where we can talk about the implementation, any teething problems, anything that's happening with it. So it means that they still have that core of expertise staying together to interact. So the team wanted to stay together, and the options there were either community practice or could they work with something else. And the team had a lot of ideas of what they could create, and what was identified was a particular, very focused short-term deliverable. And again, another prime example of what can be achieved if it's given great focus. So there was a growing trend identified by a lot of our members that health authorities were asking for additional stability testing on redundant storage sites. And this didn't, as far as every single member I spoke to was concerned, scientifically made no sense. Everyone was already testing the worst case scenario for storage. It's why would you need to test that too? It's just extra work, unnecessary testing, extra risk by having to access it. And so it was the concern was that if we don't do something about it, this might become common practice, and it might be expected and then become a regulation that you need to do it. So the group was tasked with creating a statement or position on why this is futile, why there is no need, what's the scientific justification for not doing it, and that's what they've done. So they worked over the case of, took them just over 6 months to create this position statement, which essentially is now the collective backing of all the members involved in the group saying, we believe there's no reason to do this, and this is why. And then it adds more. This is one of the other benefits of being part of the collaborations with BioPhorum, is now you have this statement which will be backed by multiple companies across the world saying this is why we believe it. So if you're ever challenged by inspectors, you can go, it's not just our belief, this is experts from across the world who believe the same thing. It has a bit more oomph and gravitas behind it. But that's again, that paper is now at the point where it's being in the process of going to go through legal review and get published. But that is, again, something that will be out there available for people to use to try and defend. And with any luck, the expectation is that it'll prevent this change coming into place and keep things as status quo rather than adding additional cost. So this in its own right is not a direct cost saving. This is preventing a cost increase in a sense. But it's a good example of what can be achieved with a short-term focus goal. And then that's finished. The group is now forming into a community of practice, as we talked about before. So again, the reason for this is rather than shutting it down, it keeps the group together. They have a lot of knowledge they wish to share and impart with each other, and also allows them to continue interacting with regards to implementation of previous deliverables. So what they have been doing is they've been taking part in various surveys on benchmarking, such as around implementation, testing strategies. They utilize Ask BioPhorum. Now this is, again, Nicola mentioned this, but any of you who are not familiar with it, this is it's a global Q&A tool that we utilize within BioPhorum to allow our members to interact any time of day around the world, to post questions and ask things that are relevant to your area of knowledge, and allows you to get feedback instantly, and then all that data is then stored and collated for future members to look at and review. So what the community is now working on is they're looking at what can be discussed. So we've got a roster of topics that's been already identified as this is what we want to talk about. And this will be put into a schedule and prioritized by the group. We've got a slide later on to show you what each of those proposed topics is about, but that's only the start. So if you are interested in joining this community, they do meet every 8 weeks, and they will, and also then we also have a continued interaction, say, via Ask BioPhorum. The team will then also, once you've joined, they'll see what the topics are discussed. If you are interested in talking about cell banks and just general best practices around cell banking, it's a good community to come and join. So if you are interested, please do let us know. Next slide then, please. We wanted to include this as well because this is for those of you in particular who may be new to BioPhorum, and this is a brief example of what we call a charter. Every work stream we have will have one of these, and it's essentially a one-page slide showing you what the team is about, what's it doing, why is it doing it, what it tends to achieve, timelines and such. So this one is a very high-level one. So this was specifically for the stability piece that we talked about, and it is. Usually there are a lot more complex, a lot more information on that than these, but this one is because it was a very focused, very targeted goal. It was quick and easy, allowed the group to move quickly because things can't. Working with people across the world and outside of your other working times, things can take some time, but this is a prime example of what can be achieved quickly. Okay, and then on to, I think, the past the ICH, go past that again, and just to give you a brief list here of the community of practice for those who may be interested in joining. This is not all of them. The team have been very engaged in putting ideas forward. This is a selection of some of the topics that the group wish to talk about in the near future. These are in the process of being prioritized by the group on order of importance for them. And then once that's done, we'll start setting a schedule for when they'll be talked about or when they'll be covered. But the idea will be that each of these will be discussed at some point in length, and members will know in advance what it is so you can come along prepared with references, case studies, anything you want to talk about, or any particular questions you want to ask about that at those communities of practice. Okay.
Thank you so much, Sean, for that introduction. Again, as Sean has said, please do let us know. There's contact details in here if you're interested in joining this work stream, but we'll take a little pause here just to check if we have any questions for Sean on cell banks. I can see we have Josiah. Please go ahead.
Yeah, thank you. I just had a question. I was wondering, are there successful examples of some of the proposed qualification approaches? Have they successfully gone through a reg authority yet? So I'm talking specifically about the figure that's included in that BioPhorum paper and around what is required for the nature of different changes. You know, for example, a like-for-like working cell bank replacement requires no small scale or commercial scale confirmation. Has that gone through successfully through a regulatory authority yet?
I think there's obviously various stages to the qualification and the levels of it. I couldn't speak to exactly which ones have been used for every stage, but obviously it has been referenced by members in previous submissions. There's obviously that mention to the Takeda, who used it as a reference and defense against the regulators when submitting the replenishment of the bank. Catherine.
Yeah, no, I just help you out a little bit there, Sean, as well. In that I know it's been incorporated into post-approval change management protocols that have been submitted to the regulators, and they've been approved. So that's the strategy that one of our members chose to take based on advice in order to integrate it into their practice. I think it's an area that we're really interested in to explore as part of the community of practice, because I think as more people, have interactions with regulators and get feedback, that then enables more confidence to build amongst the community as well.
Yeah, absolutely. I mean, certainly the proposed change scenarios and the approaches is definitely desirable. Like, looking at it, it certainly is a great state to move towards. It's always that trepidation about being who goes first, I suppose, and who has to make that case to the regulator. So that's why I was curious as to how those discussions have gone so far.
That was part of the reason why we wanted to keep the community together, is that as we have as Catherine mentioned, as our members do utilise it and get feedback, they can share with the wider group and see that there definitely are members who are going ahead and using it and doing it. So it's a matter of, it's not that everyone's sitting waiting to see who goes first. Some are already trying to do it, and it's what is the feedback we can get that they can share.
Super. Thank you very much.
Thank you.
Do we have any more questions? In which case, if there are no more questions, I will go ahead and introduce my colleague Caroline Meenan, who is going to talk about multi-product facility. Hello, Caroline.
Thanks, Nicola. Hi, everyone. I'm Caroline Meenan, Global Facilitator within Drug Substance, leading the multi-product team, which we're here to chat about today. I also have the post-approval team, and then some of the L2s on the call will be familiar with me as I lead up some of the account management as well within the forum. So specifically within multi-product, we're going to go through that today, and then the protein A resin sub-team, which is quite a large body of work for a sub-team on its own, so it gets its own wee section. If you can click across to the next one, please, Nicola. So first of all, just to give you a bit of background on the team, where we've come from and where we're kind of at now. The team have been established for quite a long time. You can see from this slide, they've got their own little library of publications. The majority of these are all focusing on cleaning, really. A few different guidelines, justifications for change, focusing on the risk involved for validation, revalidation, the different requirements and things. So there's lots of different publications there. They are all hyperlinked, and these slides will be shared alongside the recording for today. So you should have access to all of those. Where we're at now in the team, we have the topic prioritization, which I'll delve into in more detail in a minute. Looking at the day-to-day stuff that the team focus, as you can see with the library, they are very cleaning focused. The team contribute quite a lot to the annual CIP summit, which is held in the US. And what the team tend to do is they take the outcomes from the CIP summit and they feedback that into the multi-product team. We'll talk about the key learnings, key objectives, pain points, and then look at what topics we think they should focus on. So after the 2022 summit, the team came up with 4 different topics. You'll see in the middle column here, there is the droplets versus dryness. That was their first publication from the 2022 summit, and that's now available. They're working on a justification paper then for empty column cleaning, and that's currently out with the L2s for review. So if there are any L2s on the call today, you would have received the link to that technical review from me earlier this week. It is open until the end of next week. But if you do need additional time, just drop me an email or mention it to your account manager on your calls. Again, if there's any SMEs on the call, just link up with your L2s if this is an area of focus for yourself. The team has been quite a small authoring team. We've had members from Pfizer, Roche, Insight, and Regeneron. I think it's just the 4 teams working on that paper. And it'll be going then into publication and should be available in the middle of Q2. The team have identified their next paper, which is best practices for CIP skids and circuits. So that's all agreed with the L2s. Our next team call is on the 22nd of March. And within that, we're going to be calling out then for authors of that paper with a view of kicking it off. So if this is a topic of interest to you and you're not currently on the multi-product team, or you are on the multi-product team but you haven't engaged in a while, then we welcome you along. There's still plenty of time to get onboarded into the team and take part in the discussions from the outset of this paper. The team have also identified a 4th paper, which is listed in the What's Next section. They have scoped this paper out, but what they're going to do next is once the SIPs and skids is under progression, we're going to go back to the L2s and just run through the value of this paper just to make sure that the L2s are in agreement with the direction that we're going for this one. And that's quite often what you'll see when new bodies of work come up in BioPhorum. We'll always go through the L2s for endorsement. You jump on to the next slide there, please, Nicola. So topic prioritisation. This is a big one. As you've seen on the previous slide, everything is kind of day-to-day focused. What are the main pain points that our members in multi-product are incurring? What we want to do is take a step back and see, like, if we had a magic wand working in a multi-product facility, what would we really love to change? We've been working quite closely alongside our L2 sponsor, FreeJoff, and the members have contributed. It's probably running for at least the last 10 months or so, really looking into all their pain points. So what they have come up with is their overarching driver is business pressure for acceleration, maintaining flexibility and agility. And what this really means is that they want to be able to rapidly respond to global supply chain, and they can't do that at the minute. So from their brainstorming exercises, they've come up with these 4 different topics, which are outlined here. The next steps for these is going to be a deep dive into each of these, looking at what deliverables can actually come from these. We're having high-level discussions, which are going to drill down even more into the risk versus the gain, the effort required to run these projects versus the benefit that's going to come back on site. It may be things like benchmarking surveys that they want to do, which have been hugely successful in the past for all BioPhorum work streams. We've risk assessment tools outlined here. You've seen in Sean's presentation about white papers and the benefits of those. So they've no deliverables agreed at the minute. It's all at a scoping exercise, and then we'll drill down to those. So it's a fantastic time to get involved in multi-product if you're not involved. We also have quite a lot of CDMOs within the multi-product team, and they find the team invaluable because alongside the ongoing project works, the team talk quite a lot about just the day-to-day constraints that they're seeing. It's a great knowledge sharing hub. The CDMOs would experience a lot more constraints because they have their own on-site requirements alongside then the specific requirements of each of their clients. So that element of the team works really, really well. And indeed, if you are a BioPhorum member, not part of multi-product, you can ask a question through to the multi-product team. Just send that through via your account manager or your workstream lead facilitator, whoever that may be, and they can pop that across to me on the team. You go along to the next slide there, Nicola. So this is the charter for the team, just to give you a rough idea of timelines and things so you can see where we're at and what our aspirations are. There's a lot of information in here. As I said before, the slide pack will be shared. So if you have time, take a look through the charter and I'll be happy to answer any questions you may have specific on that. It's probably actually a good time to stop before I jump on to the power team, just to see, does anyone have any questions specific to multi-product for me? Okay, we'll move on to power then. The power team is significantly more complicated than the multi-product. I had the backup yesterday of one of my SMEs from Roche. So I've no SME today, but if there is anybody on the power team that wants to give a shout out, then I'll welcome your backup if there's any complex questions coming from the audience, please. This slide is really condensed, hopefully self-explanatory for you all. You can see where the current state is within industry. So the chromatography resins are dedicated to single-use products. They have limited manufacture. The resin use is not being fully utilised. So we do have sustainability elements here. The reuse of resin for multi-products isn't considered for GMP, for API. There's no clear regulation, so you can see kind of the driving point from the team before we even look at cost points and things. Protein A's then are widely used for the manufacture of MABs and the therapeutic protein products. So this is why we selected the Protein A to kickstart the team. I know there are different resins used, but just to give you an idea of the drivers for this project. So moving forward, what the team want to do is demonstrate that the Protein A resin reuse is feasible when moving from product A to product B, maintaining the critical performance attributes and the critical quality attributes of the API. So this project has been running for quite a long time because there's so much work involved in it to demonstrate this. You will note at the bottom right-hand corner of the screen, we've outlined the University of Limerick and BioPhorum. So the University of Limerick are doing a similar project in a sense that they are looking at the reuse of Protein A resin moving from product A to product B. However, their studies are theoretical. They're doing a lot of development, so their work complements BioPhorum's work, and we have a memorandum of understanding in place between the two of ourselves, and we're working quite closely. They will come along and present their findings to BioPhorum, and they would also review ours. So that partnership is invaluable to the team, and we really do complement each other, and it's great to have that additional resource to rely on. The impact of not being able to use the resin is the costs, logistic challenges, storage. If you note here in the bold at the bottom, there is significant underutilization of the Protein A resin for the clinical manufacturers compared to commercial products. And again, it raises a lot of questions with regards to sustainability, which is one of our key value drivers in the team. The benefits, the cost benefit speaks for itself. You can clearly see here up to $8 million per site in the reduction of manufacturing costs. So when I spoke previously about the topic prioritisation, and we said that we really need to look at the value and what benefit that's going to bring back. The power sub team, when that was initially being scoped out, it was a non-runner really when you look at the cost benefits that can bring back. On top of that, you've reductions in resource, reduced storage and testing costs, and then the security of supply as well there. So huge amount of information on this slide, but it should be quite self-explanatory as to why we are where we are. On the next slide, we've outlined where we're at so far and where the team is going to go moving forward. So those highlighted in green are all complete by the team. We've done lots of literature reviews, a lot of background work into what's been done before. Our member organisations have presented case studies back to the team. I'll grab your question at the end of this slide. Thanks, Mama. And those case studies are available for members of the multi-product team. Most of them, a few of them have legal restrictions on those. So we haven't shared those slides, but the members are still in the team. So if we do have questions, we can still reach out to them. A lot of the literature that we have reviewed as well has been condensed into a nice little catalogue. And you'll see even on the team's FMEA, we are frequently referring back to that literature review. And it's great to see some of the BioPhorum members actually contributing to that literature also. At the minute, the team are talking about exposure limits. We have hopefully locked those down, and we're ready to progress the scoring of the FMEA within our member organisations. We have a small kind of ad hoc team really working hard in the background from Lily, Roche and Regeneron. So they're really trying to push things along in the background. We've had some great new SMEs added onto the team, and we're really challenging what's been done so far. So there's a fantastic body of work being carried out already. Moving forward then, as you can see in the Navy, they are going to move on to the risk assessment paper, the white paper, and then the publication and socialisation of that. I've included the charter as well, just for your reference on the next slide, so people can see the timelines of where things are at. I'll just take a look at your question now.
I was going to say, hi, Pam, did you want to go ahead and ask the question?
Thanks, Nicola. Caroline, I haven't been close enough to this work stream, so apologies if this is an obvious one. I'm assuming this team are also looking at the impact to resin lifetimes if they're talking about reusing across multiple molecules.
Yeah, and that forms part of the FMEA, and where the team are at the minute is they're going back and revisiting the FMEA. Sometimes they find it quite challenging because they often drift back into what happens when you're doing product A to product A. So they're moving back there and they're really focusing on the product A to product B. What would the difference be on the resin lifetime if you were changing from product A to product B?
Thanks, Caroline.
Does that answer your question, or do you need further information?
No, that's perfect. Thanks.
Thanks, Mama.
Thank you so much for that, Caroline. Great job as well explaining protein A resin reuse. Like it says on the slide, and as Caroline has already said, you're very, very welcome to join either multi-product facility or the protein A resin reuse team if you're not already involved here. And just to check if we have any final questions on either of those teams before we move on. Okay, that takes us on to our final work stream for today, and I will invite Sean back to tell us about the LIVCA team. Hi, Sean.
Thank you again, and hello once again, everyone. So LIVCA, as a work stream, this was very much born out of the same idea. The idea of this was born from the back of the Cell Banks paper. Because the process, obviously, Cell Banks Group was created and made the paper based on a lack of guidance, not a lot of information on exactly how it should be performed, and very much down to individual company interpretation. LIVCA is essentially the same situation. It's something everyone has to do. It's obviously something very important, but the guidance that exists out there is quite limited. And so the thought was, could we do something similar with LIVCA? Could we identify a best practice guide on this is how to perform LIVCA studies to try and streamline and minimize that? Put on my next slide, please, Nicola. Oh, that was thought we had the other one.
Did you want your next slide? Sorry.
Yeah, I thought we had the chart on first, just to put it there. So this is, again, I'm not going to speak to this in great deal. It's there for you to review afterwards, but really this sums up what the LIVCA group was about. It started off, again, with the idea of it being LIVCA studies are complicated. LIVCA studies are, we all know that, and they are complicated and work intensive and costly and overall just a burden that ideally could be addressed. And what we wanted to the team came about the idea of, can we create a best practice and work from there? So the first task of the group was then to essentially create a benchmark of what everyone does currently. So they made a comprehensive survey on what do people do for LIVCA right now, and whilst also using the opportunity as part of that data gathering exercise to understand what other aspects people do, for example, things like small scale, because the thought process with this was LIVCA is a process that is typically done on a commercial scale, at commercial scale. But a lot of aspects of it, at least by our members' belief, is that you could justify it easily at a smaller scale, either pilot or potentially smaller. That is not, but it's not generally accepted by the industry. A lot of the guidance out there would often reference potentially using pilot scale, but the understanding of most members was that whenever you did this, you typically got refused by the regulators, and they expected it to be done at full scale, even though the guidance said you could do it. So one of the key tasks of the survey was to understand what are people actually doing. Is anyone actually using small scale, as well as understanding what your practices are? What approach do you take to defining LIVCA? And then beyond that, the expectation was we create a paper like that. So this, again, a good example of what the charter defines. It has a lot of information in there, but again, I'm not going to go through that in detail now. So if you want to jump back for a second, Nicola. So what we achieved, this survey, as I mentioned, was put out there. It was quite comprehensive. There was a lot of information in there, a lot of questions, and an awful lot of data from what to process. And what it actually showed was kind of what we knew already, but not actually that helpful, was that LIVCA is very complicated, and there is no one simple way to do it. There is no simple one best way to perform LIVCA. What was the main takeaway from it? It is that there are multiple different approaches that companies have taken, which are quite different, even in how you actually define your LIVCA. That was a big kind of crux, was how do you actually define what starts and ends of your LIVCA, and this then caused problems on how can we can't then create a simple best practice guide. So the group then took that as an action to okay, let's boil it down. What are a few best strategies to perform LIVCA, and what are the pros and cons of those? And so that was then decided what the paper would focus on. Create a guide on performing LIVCA, because again, right now the guidance out there is very, very minimal. And I know that already from what we've been prepared so far, a lot of members think this is going to be beneficial for them in helping them plan future LIVCA studies. It's about defining what should and shouldn't be done, and what aspects of it are riskier, because depending on where you want to define it, where you want to do your testing, what aspects are adding risk or taking away risk. But we didn't think that was enough to just look at defining LIVCA, how to do it. It's, yes, it'll be a helpful guide for everyone, but we wanted to go beyond that. In particular, looking at what new technologies or new testing procedures could be utilised to try and improve or bring down costs and things associated with performing LIVCA studies, as well as exploring small scale opportunities. Because as I mentioned, a lot of the members who are taking part in this believe scientifically, a lot of it can be a lot of the aspects of LIVCA can be justified at a smaller scale, minimizing your costs. And so that's one of the aspects and the focuses of the paper, is to kind of look at what can be done about that. Now, we had one of my SMEs in particular highlighted one of the key things to take away from it is there has been successful companies that we know of who've used small scale, not even pilot, to conduct LIVCA studies, and this is referenced and included in the paper, the fact that they've done this even at bench scale, and it's been and had approvals through the agencies. But one thing to consider with that, and what is also discussed through the paper, is small scale isn't necessarily always the best option, because by the time you've then done other aspects to it, and qualified it and made sure it's equivalent to a large scale, you might as well have worked at pilot, which is still better than commercial, but that is something to consider. So we are, and that is one of the aspects that I discussed about in this paper, is that's what this team's looking at the different ways that we can improve performing LIVCA studies, making them more streamlined, making them quicker, and making them more clear, and whilst also exploring the options available to members. So the intent of this is to put down, these are the scientific reasons behind it, these are the risks associated with it, so each individual company can then make their own informed assessment based on an increased amount of guidance that comes from this paper. And so right now, the group are working on this. They've been working on it for a good number of months now. They're very much in the late stages of drafting it. It's almost complete. They're expecting to have they're planning anyway to have the finished draft completed within a month. And that will then ideally be looking to get published sometime within Q2 next year. Sorry, this year, not next year. Q2 this year, not next year. And then that'll be available for you to see. So what could be, and then we're talking about what could the group be doing next. The paper very much focuses on the CHO cell line, because one of the was realised when we did the original survey is the majority of our members work in CHO. There is obviously other cell lines available that people do work in, such as microbial insect, but how you perform LIVCA and how you are doing that is quite different depending on the cell line that you're operating in. So the group has decided, the biggest chunk was CHO, because that's the majority of members' cell lines. So that's what we work on first. And depending on how it's received, the group would be keen to explore looking at what could be done with microbial insect LIVCA. How could you adapt this existing paper we've got to see what could be done for different cell lines and to see how relevant it is. So if you are interested in either of those future, it's potentially a good opportunity to get involved. There's no guarantee we will be going ahead and doing that, but it's just that's something that the team are keen to do, and it'll all come down to, again, the value of it, because as I say, if there's not a lot of people doing microbial insect, it's a lot of resource to attribute to it. But the team are very keen and very passionate, and there's an exceptional amount of knowledge within the team. I'm always blown away. Like, I myself, I'm not a large molecule expert, and half the time I'm listening to them and listening to their expertise, and it just blows me away, some of the stuff that they come out with. But beyond that as well, we also want to follow a similar path that we did with the cell banks, in that this is another situation where there's little guidance that exists. So we're proposing essentially a set of a process in order to do it. So we want to get out there and make sure that it's approved and accepted by the community and by regulators and the like. So what we want to ideally do is take it out to conferences as well, present this is what we are suggesting as the way LIVCA should be done. This is why I believe utilising different technologies and smaller scale can be beneficial. So we want to, that'll be the next steps once the paper is published, is get our members out to conferences, spreading the word, socialising it, in the same hopes that, again, once again, we can hopefully try to influence the guidance that doesn't particularly exist and say, this is how we think we should go about doing it. And with that, I mean, that's the question. So if anyone does know of any particular conferences that you're aware of that you think this might be beneficial to, please let us know. And yeah, so this group is still, I say, it's towards the end of its time with regards to this paper, but it's still got a lot of work to do, as I say, with regards to socialising, implementation, getting it beyond that, and potentially looking at future topics. So it's still a good time to get involved with this group if you are interested in LIVCA. It is obviously a very focused and kind of specialised area, but it is still something that is critical for all manufacturing. And so if you are interested, let me know. They meet every 3 weeks, and it's quite a good team.